Okay, so we shall continue with the next chapter under autocoids. We had started with the next chapter in your syllabus, that is autocoids, under which we have already covered the histamine and the antagonist. Okay, so we shall continue with the next chapter, that is serotonin, which is also known as 5-HT or 5-hydroxytryptamine, and the antagonist of this serotonin or 5-HT antagonists as they are called as. Coming to the outline of the chapter, we shall cover the synthesis, storage and destruction of the serotonin, then the serotonergic receptors in brief, the actions of these serotonins on different systems of the body. Next part is the pathophysiological role, that is pathological and the physiological roles which serotonin plays in our body. Next comes the uses, then the drugs affecting the 5-HT system. So what are the different drugs which can alter this 5-HT system, maybe synthesis or maybe release, maybe the uptake, okay, or the destruction. So likewise, what are the different drugs which can alter the actions of this 5-HT system. And the last part in the chapter is the 5-HT antagonists, which will be covered in detail. So which are the drugs which can antagonize the actions of 5-HT at the 5-HT receptors. Okay, so to start with the introduction, now this serotonin, okay, when it was discovered way back in 1990s, before 1950s. So at that time, two different studies were carried out. Okay, so parallelly two studies were going on, and in one study the name was given to a particular the chemical which was found in the body. Okay, so the substance which was found in the body, okay, and it is supposed to produce, or it used to produce the vasoconstriction. Okay, and it basically was found in the serum when the blood clotted. After the blood clots, okay, in the serum this particular substance was found, and was found to produce the vasoconstriction, the constriction of the blood vessels. And the name given to this particular chemical was serotonin. Okay, because it was because it was found in the serum. And in another study, it was found that one particular substance, it produced the contracting action on the smooth muscles. So smooth muscles could be contracted by this particular substance, and basically it was located in the enterochromaffin cells of the gut mucosa, that is in your GI tract. Okay, so that's why the name given to this substance was enteramine. Okay, so these were the two different names given depending upon where exactly they were found, or where exactly those chemicals were located. Okay, but later on in 1950s, it was found that both these substances was the same. It was found that it was the same substance, and it was in this particular year, both these substances which were earlier named as serotonin and enteramine, later on it was named as 5-HT, that is 5-hydroxytryptamine. Okay, now it is well understood, it is studied in detail, this 5-HT, the structure, synthesis, degradation, its distribution in the body. It is very well understood, and it is found that almost about 90% of the body's 5-HT, it is mainly located in your intestines, and the remaining 10%, it is found mostly in the platelets and the brain, where it acts as a transmitter. So this is how it is distributed in the body. Now apart from this, it is also found in the wasp and scorpion sting also, some insect bite, okay, that also can release this 5-HT at that particular site. And it is also widely distributed in the invertebrates, and even it is found in the plants also, like banana, pear, pineapple, tomato, stinging nettle and cabbage. Okay, so it has a widespread distribution, this particular serotonin. Okay, so coming to the first part, that is the synthesis, storage and the destruction of this 5-HT. Now basically, if you can see the structure of this 5-HT, this is the structure of your serotonin or 5-hydroxytryptamine. So it is the chemical name of this 5-HT, it is beta amino ethyl 5-hydroxy indole. Okay, so at beta position there is an amino ethyl chain, and at fifth position there is a hydroxy group. It's a indole. Basic what is it? The basic structure is the indole, and there are certain attachments. So accordingly, it is named as beta amino ethyl 5-hydroxy indole. Now the synthesis and the destruction part is discussed in this particular picture. Okay, so coming to the synthesis part, it is synthesized from tryptophan, and you all are all aware that tryptophan, it is an amino acid which is normally found in the body. Okay, so it will have a carboxy terminal and it has an amine terminal also. So from this tryptamine, with the help of two step reaction, 5-hydroxytryptamine is generated. Okay, now this tryptophan, it is acted upon by hydroxylase. Okay, this is the enzyme which acts upon tryptophan, and this tryptophan gets converted into 5-hydroxytryptophan. So a hydroxy group is introduced at the fifth position with the help of this enzyme hydroxylase. So one OH group is introduced on this tryptophan at the fifth position. So tryptophan gets converted into 5-hydroxytryptophan. Further, this 5-hydroxytryptophan, it is acted upon by decarboxylase. Okay, so an amino acid, it has a carboxy terminal also, COH group is there. Okay, so that group is removed with the help of this enzyme decarboxylase, and this 5-hydroxytryptophan gets converted into 5-hydroxytryptamine. That COH group is removed over here. Okay, so that's how it gets converted into 5-hydroxytryptamine. Okay, so this is how the serotonin is generated. Then coming to the destruction or the metabolism of 5-HT, it is acted upon by enzyme MAO. Monoamine oxidase is the enzyme involved in the metabolism of 5-HT. Okay, and it gets converted into 5-hydroxyindole acetic acid. Further, it can be also acted upon by dehydrogenase, and it gets converted into 5-hydroxytryptophol. Okay, so there are two metabolites which are generated from the metabolism of this 5-HT. Now coming to the storage part, okay, or the uptake part. Okay, now this picture you all must be familiar of. We have seen this picture under the chapter antidepressants, right? Most of the antidepressants, we are all aware that they act by inhibiting the reuptake of noradrenaline and 5-HT. Okay, so here in this figure it is shown that the Na, which is released, it is taken up back by the specific active transporter which is located on the axonal wall, that is norepinephrine transport. Okay, and further, once it reaches the exoplasm, from here it enters into the storage vesicles. It is basically stored in the membrane bound vesicle. So here also there is an active transporter. Okay, so that is vesicular monoamine transporter 2, which is responsible for uptake of this Na back into the vesicles. Now in the same way, similar process is also utilized by the 5-HT also. Okay, so just like for Na, there is a transporter, norepinephrine transporter. The same way, for the uptake of 5-HT, there is a transporter known as serotonin transporter, SERT, it is known as. Okay, so this is responsible for the uptake. Okay, so whatever circulating 5-HT is generated, it is taken up by the cells. Okay, or the serotonergic nerve endings, okay, or the platelets. They take up this serotonin with the help of these transporters, and it is stored inside the vesicles. Okay, it is utilizing this transporter also. Okay, vesicular monoamine transporter 2. Okay, same one which is utilized by the noradrenaline. The similar type of transporter is also utilized by the 5-HT. For it to get stored within the vesicle. So just like over here, noradrenaline is getting stored in the vesicles, in the same way 5-HT is also stored within the storage vesicles. So process is similar to that of the catecholamines. Okay, noradrenaline is one of the catecholamines we are all aware of. Okay, so similar process is followed by this. Even the metabolizing enzyme MAO, that is also same for both. Okay, 5-HT is also metabolized by MAO, and noradrenaline also is metabolized by enzyme MAO. Now here for 5-HT metabolism, MAO-A, we have seen that there are two subtypes of this MAO, right? Okay, so preferably this MAO-A enzyme subtype is responsible for metabolism of the 5-HT. Okay, so this to do with the synthesis, storage and the metabolism part. Then coming to the serotonergic receptors or 5-HT receptors. So these receptors, they were discovered by these two scientists, or they were classified basically into two types by Gadam and Picarli in 1957. They classified these 5-HT receptors into two types basically. Okay, so these two types are musculotropic and neurotropic receptors. Okay, and this musculotropic receptors also are known as D type of receptors, and neurotropic receptors are also known as M type of receptors. Okay, now why these are named so, why this musculotropic is known as D type and why this neurotropic is known as M type is because of the blockers. Okay, it is named as D and M depending upon the blockers or the antagonists at these two types of receptors. So basically this musculotropic 5-HT receptors, these were blocked by dibenzylline. Okay, so D for dibenzylline, so that's why they are known as D type. And the neurotropic 5-HT receptors, basically they were blocked by the morphine, that's why they are called as M type of 5-HT receptors. Okay, so again there are certain agents, examples they have given, methysergide and cyproheptadine. Okay, so these are also the antagonists of the 5-HT. D type of receptors are blocked by these antagonists. Okay, so now there are number of 5-HT receptors which are discovered. Okay, so all of these 5-HT receptors, they have been cloned and it has been well classified into different types. Okay, so basically they are divided into 4 families. Okay, so certain receptors are grouped together, like the 5-HT1 is categorized separately, 5-HT2, then the 5-HT3, and these 5-HT4 to 7, these are grouped under the same family due to the similarity in their actions. Okay, so they are categorized under the same family. So basically these 5-HT receptors are divided into 4 families: 5-HT1, 5-HT2, 5-HT3 and 5-HT4 to 7. Okay, now again within these types of 5-HT receptors, again there are subtypes like 5-HT2A, okay, 5-HT2B. Okay, depending upon the subtypes, further subclassification, a letter has been assigned to them. Okay, we will come to know in the description, but all of them are not that important or clinically significant. Okay, if you consider all the 5-HT receptors, there are totally 14 types of these 5-HT receptors. Okay, so again over here there are subclassification. These are totally 7 5-HT receptors, but further there is subclassification again within this. Okay, so totally there are 14 subtypes of 5-HT receptors which have been discovered. Okay, now coming to the transducer mechanism. Now what is the mechanism by which they transduce that particular their signal once they are stimulated, how they carry that particular signal, or what exactly is responsible for production of the action. Okay, so if you can see that all the 5-HT receptors except 5-HT3, okay, so far we have seen that there are 14 subtypes of 5-HT receptors. So all of them except this 5-HT3, all are G protein coupled receptors. You are already aware of the transducer mechanisms, right? Okay, so all of them except 5-HT3, these are G protein coupled receptor. Now within these also, again, the way by which the signal is transduced, that is again different, because we are already aware about the different types of G protein coupled receptors also, right? Depending upon the transducer mechanism, again these G protein coupled receptors are also different. There are different pathways which is followed, right? So if you can see that is also different among these receptors, okay, all of which follow the G protein coupled receptor mechanism. Again, in that also there is variation how that signal is transduced. Okay, so this 5-HT1 receptor, it basically acts by decreasing the CAMP production. Okay, you are already aware of there are different pathways, right? IP3-DAG pathway, phospholipase C IP3-DAG pathway, adenylyl cyclase CAMP pathway, and channel regulation. Okay, so these are the three ones under the G protein coupled receptor. So out of that, this 5-HT1 receptor acts by decreasing the CAMP production. Then next 5-HT4, 5-HT6 and 5-HT7. These receptors, they basically act by increasing the CAMP production. And the 5-HT2 receptor, it basically acts by generating the IP3-DAG as the second messenger. Okay, so there are different pathways which are utilized by different types of receptors. Now over here we have seen that all the 5-HT receptors except this 5-HT3, all are G protein coupled receptor, and this one, that is 5-HT3 receptor, this is a ligand gated cation channel. Okay, so this is not a G protein coupled receptor, it's a ligand gated cation channel, and sodium and potassium ion channel it acts as, and stimulation, it is responsible for the depolarization. So this is to do with the transducer mechanism. Coming to the actions. Okay, now this particular 5-HT, it's a potent depolarizer of the nerve endings. Okay, and so also if you can see the overall actions, sometimes it exerts direct action. Okay, sometimes at the same site it exerts the action reflexly. Okay, maybe it produces indirect effects. Okay, so this complicates the picture exactly over the action at particular system. Also tachyphylaxis is quite common when repeated doses of this 5-HT are given. Tachyphylaxis meaning the effectiveness reduces over a period of time. Okay, so because of this the overall effects are often variable, if you can see. Okay, now coming to the actual actions, the first one is on the CVS or cardiovascular system. Okay, now over here also the actions are quite widespread and quite variable also. Okay, so coming to the actions on the arteries. Now arteries are constricted as well as dilated. Okay, so both is possible. Constriction of the arteries is also possible and dilatation of the arteries is also possible. Now let us see how or by what mechanism they can be constricted. Okay, this constriction, it is basically because of the action of 5-HT on the smooth muscle. So directly it will produce the action on the smooth muscle, it will produce a contraction of the smooth muscle, and it will result in the constriction of these arteries. Okay, now again it produces the dilatory action also, it can dilate the arteries also. Now this dilatation, the dilatation of the arteries is basically because of the release of the EDRF, endothelium dependent relaxing factor, that is nitric oxide, NO is released, and that is responsible for the smooth muscle relaxation. So obviously because of the smooth muscle relaxation in the arteries, it will result in the vasodilatation. Okay, so this is to do with the actions on the arteries. Now again the action will depend upon the vascular bed and the basal tone of the smooth muscles. Again it will be variable depending upon where exactly these arteries are located or what is the basal tone in these smooth muscles of these arteries. Okay, then in addition to this, 5-HT also releases the ADR from the adrenal medulla. Adrenaline release is also enhanced by 5-HT from the adrenal medulla. We are all aware of that adrenal gland, the adrenal medulla, the central part. it produces the ADR. So the release from this adrenal medulla of the ADR is also enhanced. So this in turn will affect the ganglionic transmission and will evoke the cardiovascular reflexes. Okay, so it will act on the ganglionic transmission. It will be stimulated, the sympathetic ganglia. Okay, their actions will be enhanced and cardiovascular reflex will come into picture. So if you can see, the overall actions are quite complex. Okay, so net effect is quite complex. Okay, you will never know what will happen exactly in the arteries at one particular place in the body and in the another particular area in the body. Okay, so it's quite variable. Okay, apart from that, if you can see the actions on the larger arteries and veins, these are characteristically constricted. So here no dilatation is seen, only the constriction, vasoconstriction is seen in the larger arteries and the veins. So we were talking about the smaller arteries over here. Okay. Apart from that, in the microcirculation, that is at the arteriolar or venule levels or capillary level. Okay, here it dilates the arterioles and constricts the venules. Okay, so if you can recall your circulatory system, from left ventricles, the blood is pumped into the aorta. It goes into further the smaller arteries. Then from there it will go into the arterioles. Next it will go into the capillaries. From capillaries the blood will be taken into the venules, then comes the veins, then the superior inferior vena cava, and the blood comes back to the right hand side, atria. Okay, so this is your circulatory system. Okay, now what 5-HT is doing in the microcirculation, that is at the smaller blood vessel level, arterioles are dilated and venules are constricted. Okay, now because of the dilatation of the arterioles, there will be increase in the blood flow in these arterioles. Obviously the blood flow will increase. Wherever dilatation occurs, the blood flow to that area will increase naturally. Okay, so because of this dilatation of the arterioles, blood flow over there will increase. The capillary pressure will increase. Okay, because there is more blood pouring in that particular area. Okay, so because of the rise in capillary pressure, obviously we are all aware that under CVS we have seen that at capillary level filtration takes place, right? Okay, so there is a lot of exchange of material which is taking place at the capillary level, and because of the pressure changes, there is also filtration which is taking place. That is, except your plasma proteins and the blood cells, your plasma can be filtered out. Whatever dissolved material is there, it can come out of your blood vessels, because capillary wall, it is made up of only the endothelial cells, and that too are placed quite apart. There are gaps in between, they are not held tightly. So because of rise in this capillary pressure, fluid also will escape out. Okay, so this is what is happening at the microcirculation level. Okay, then coming to the actions on your heart, if you can see the isolated heart, okay, the one which is isolated, okay, in vitro. Okay, so on the isolated heart, 5-HT produce the stimulant action. It is stimulated by 5-HT. Now this stimulation of the isolated heart is both produced by directly and by release of the NA, noradrenaline, from the nerve endings. Okay, so both are responsible for the stimulant action on the heart. Now in intact animal, okay, so this is what is happening in the isolated heart. Now in intact animal, bradycardia is seen mainly, that is the decrease in the heart rate. In case of isolated heart, it was getting stimulated, that is the heart rate as well as the force of contraction was increased. But here in case of intact animal, bradycardia is seen, exactly opposite effect, that is the decrease in the heart rate is seen. This is because of the activation of the coronary chemoreflex, which is popularly known as Bezol-Gerish reflex. Okay, and this is mainly mediated through the vagal nerve, which is supplying the heart. Okay, so because of this, it will result in bradycardia, hypotension, that is decrease in the blood pressure, and apnea. Okay, that is cessation of the breathing. Okay, breathing won't be appropriate. Okay, so these are the consequences in the of the 5-HT on the heart of an intact animal. Now coming to the actions on the BP. Now actions on the BP is a triphasic one. A triphasic response is produced, okay, upon the IV injection of this 5-HT. So initially effect will be little bit different. Later on it will change the actions, and again prolonged action will be slightly different. Okay, so if you can see, there will be an early sharp fall in BP. Okay, so initially when you inject this 5-HT by IV route, initially it will result in a sharp fall in BP. That is, BP will fall. Okay, and this fall in BP, that's basically because of this coronary chemoreflex. Here we are seeing hypotension is produced. Okay, so this early sharp fall in BP, that's mainly because of this coronary chemoreflex. Okay, then later on, a little later, there will be a brief, that is a short lasting rise in BP will be produced. Now this short lasting rise in BP is because of the vasoconstriction and increase in the cardiac output. We have seen an actions on the blood vessels is it produces vasoconstriction, right, of the smaller as well as the larger blood vessels. So that is responsible for the rise in BP. So also we have seen that it has the stimulant action, also cardiac output may be also increased. Okay, and in case of the intact animal, actually it produces the bradycardia. So the reflexes will come into picture and it will start stimulating the heart, and cardiac output may be increased. Okay, then a prolonged fall in BP is seen later on. Okay, after these two phases, a prolonged, that is a long lasting fall in BP will be seen, and this is mainly because of the arteriolar dilatation and extravasation of the fluid. Okay, we have seen that arterioles are dilated, okay, and extravasation of fluid meaning the capillaries, from the capillaries more and more fluid will come out, right, because of the arteriolar dilatation. Okay, the more and more blood will come over there, capillary pressure rise will take place, and the fluid will come out from the capillaries. So the volume of blood is reduced, so that is responsible for the fall in BP. So this is how a triphasic response is shown by the 5-HT. Initially a sharp fall in BP, after that a brief rise in BP, and later on a prolonged fall in BP will be seen. And we have already seen what are the reasons for this triphasic response which is produced by 5-HT. Okay, but as such it does not have any role in the physiological regulation of the BP. Okay, normally BP regulation, 5-HT does not play any role. So this to do with the action on the CVS. Next, coming on the actions on the visceral smooth muscles. Okay, in the smooth muscles of your visceral organs. Okay, basically it affects only two organs, two visceral organs. Okay, the first one is your GIT. There are smooth muscles in your GIT. So 5-HT, it is a potent stimulator of the GIT. That is the smooth muscles in the GIT. These are stimulated, okay, both directly by a direct action as well as through the enteric plexus. Enteric plexus, it includes the nerves which are supplying to your GIT. Okay, so it will stimulate your GIT or contract the smooth muscles in your GIT, both by directly acting on the GIT, because there are several 5-HT receptors which are located on your gut wall. Okay, so there are so many 5-HT receptors. So either 5-HT will go and bind to these receptors and stimulate the contractile property of your GIT, or they will act indirectly through stimulation of this enteric plexus. Both the ways it will result in the stimulation of the GIT, that is, it will increase the contractile property of your GIT. So peristaltic movement will increase. It will increase the peristalsis, and diarrhea can occur because of the action of 5-HT. So this is to do with the actions in the GIT, and also we have seen that more than 90% of your 5-HT, it is found in the GI tract. Okay, then one more visceral smooth muscle which is affected is the one which is located in your bronchi. Okay, bronchial smooth muscles are also contracted, because of which the bronchoconstriction will be produced. Okay. okay, under the action of 5-HT. So it can be quite dangerous in case of asthmatic patients. Okay. However, this bronchoconstriction which is produced by 5-HT, it is less potent as compared to histamine. We have seen under the actions of histamine, histamine is also a potent bronchoconstrictor. Okay. So, but 5-HT is comparatively a less potent bronchoconstrictor when you compare it with the histamine. If you can see the actions on the other smooth muscles, it is feeble and inconsistent. Very little action is produced on the smooth muscles of the other visceral organs. So it is not quite significant. Only GIT and the bronchi, smooth muscles, are mainly affected. Contraction is produced so that diarrhea or bronchoconstriction may be precipitated. Next is the action on the glands. 5-HT basically inhibits the gastric secretion. Okay, that is acid and pepsin which is generated and secreted in your stomach. The secretion is inhibited by this 5-HT. Okay, and simultaneously it is also increasing the mucus production. Okay, so you are all aware about the peptic ulcers, right? So in that we have seen that the imbalance in between this protective and aggressive factor is actually responsible for precipitation of the peptic ulcer. Okay, so in peptic ulcer what happens is aggressive factors, they are exaggerated, like say maybe the acid production is increased, okay, and the mucus production may be reduced. Okay, so if you can see the effects of 5-HT, what it produces, it inhibits the gastric secretion. So acid secretion and pepsin secretion is inhibited by 5-HT, and at the same time it is also increasing the mucus production. So the aggressive factors are suppressed or inhibited by 5-HT, and they are enhancing the protective layer. Okay, mucus production, it produces a protective layer so that your stomach mucosa does not come in direct contact with your acid. So it forms a protective layer, and when this mucus layer is disrupted, acid will come into contact with your mucosa and it will start damaging it. Okay, so that's how 5-HT it has a ulcer protective property. Okay, so in the GIT, that is basically in your stomach. As such, it does not produce any significant effect on secretion of the other glands. Okay, mainly it affects the secretions in your stomach. Okay, the next action is on the nerve endings and the adrenal medulla. So coming to the actions on the nerve endings, efferent nerve endings, these are activated. Okay, so it will result in the tingling sensation or pricking sensation. Okay, as if someone is pricking you with a needle. Okay, or painful sensation may also be produced because of the stimulation of these efferent nerve endings. Okay, so these efferent nerves, they are acting as the receptors. Okay, so since they are stimulated only when there is some kind of stimulus, maybe internal or external stimulus. But here 5-HT is activating these nerve endings. Okay, so that's why these effects are produced. Okay, then depolarization of the visceral efferents. Okay, in your viscera, those nerve endings which are supplying to your visceral organs. So stimulation of these visceral efferents, okay, that is responsible for either respiratory and the cardiovascular reflexes, or nausea and vomiting may also be precipitated. Okay, then 5-HT also it can release the catecholamines. Coming to the actions on the adrenal medulla, we have seen that histamine also can release the catecholamines, but 5-HT comparatively it is a less potent one in releasing the catecholamines from the adrenal medulla. Next is the action on the respiration. Okay, so here a brief stimulation of respiration and hyperventilation may be seen. Okay, and in larger doses, transient apnea may also be seen. This is basically because of the coronary chemoreflex. Okay, then next, coming to the actions on the platelets. Okay, now here 5-HT it changes the shape of platelets. Shape may be altered, and it is a very weak aggregator of the 5-HT through this 5-HT2A receptor. I told you that there are subtypes also. So 5-HT2A basically has this particular action. platelet aggregatory property. Okay, so when 5-HT is stimulating these receptors, it will result on the platelets basically. So it will result in the platelet aggregation. Usually platelet aggregation, it occurs whenever there is any damage to your blood vessels. Okay, so it plays a very important role in your clot formation, platelet aggregation. So 5-HT also is responsible for this platelet aggregation, but it is a weak aggregator. So it has a weak platelet aggregatory property. Okay, as such, release reaction is not induced by this 5-HT. Only it can change the shape, and very weak aggregator it is. The next, coming to the actions on the CNS. Okay, as such, this 5-HT, it can cross the blood-brain barrier poorly. Okay, it cannot cross the blood-brain barrier very effectively. So that even if you inject it by IV route, it does not produce any central effect. Okay, however, normally this 5-HT, it acts as a transmitter in your brain. Serotonergic neurons are there. Okay, so it will act as a transmitter. Basically it is an inhibitory transmitter. Whereas if you can directly inject this 5-HT in your brain, it can produce actions like sleepiness, changes in the body temperature, hunger, and a variety of behavioral changes may also be produced. Okay, but as such, when you inject it by IV route, it will not be able to produce any CNS effects because it cannot cross the blood-brain barrier. Okay.